Hello, everyone. Thank you all for joining us today, and welcome to the Monarch Butterfly Conservation Webinar Series. Today's webinar is titled Insights from 50 Years of Data on Monarch OE Infection. My name is Katie Lynn Bunny, Education Manager for the Monarch Joint Venture. We're excited to continue hosting this webinar series with the U.S. Fish and Wildlife Service's National Conservation Training Center. We are delighted to host this presentation from Ania Majewska, Assistant Professor at University of Georgia. And for this webinar, we're curious where you're all joining from. If you're comfortable, please share in the chat where you're located. Before we get started, here are a few expectations and community guidelines. Community guidelines for the webinar. During the webinar, please put your questions in the chat box and we'll collect those for the Q&A session at the end. And just a reminder to keep the chat friendly and professional. Thank you in advance. If you pause or click away or refresh the page while you're viewing the webinar, you will need to click the play button again. It will start where you left off. If you want to catch back up, you will need to slide the bar at the bottom of the video window. This webinar will be recorded and posted on both the NCTC and Monarch Joint Venture websites in 2 to 3 weeks. The entire Monarch Conservation Webinar Series archive is free and available to watch and contains about 7 years worth of Monarch Conservation webinars on both of our websites. If you're joining us today, but you didn't pre-register for the webinar, we would love to count you for attendance and be able to send you some follow-up resources. So please email education@monarchjointventure.org with your name and email address. And finally, all of our webinar speakers represent their own viewpoints, which are not necessarily those of NCTC or the Monarch Joint Venture. And now I will introduce our presenter. Ania Majewska is an assistant professor in the College of Veterinary Medicine at the University of Georgia. Despite growing up in a concrete jungle, Warsaw, Poland, Ania has been a nature enthusiast for as long as she can remember. Her passion and curiosity about nature led her to pursue a bachelor's degree in biology, a master's in wildlife biology, and later a PhD in ecology. Her PhD research focused on understanding how the decisions we make in gardens in terms of plant selection and management can benefit or harm insect pollinators. Additionally, she studied how non-native milkweeds impact monarch butterfly biology and the incidence of disease in monarchs. Ania lives in Athens, Georgia with her family and enjoys running, hiking, gardening, and beekeeping in her free time. Now I will turn it over to Ania.
Wonderful, thank you. Let me just go ahead and set up my screen here.
There you go.
Awesome, perfect. All right, well, thanks so much for the invitation to share some of the research I've done as a graduate student and later as a postdoc. So I completed my PhD with Dr. Sonia Altizer and my postdoc with Dr. Jaap de Rood, and I will again be discussing some of the work and the things I've learned over time about the monarch and its parasites. So a roadmap of the talk that I'll be giving. First, I'll tell you about OE, the parasite that monarchs are associated with. I'll tell you a little bit about how it impacts monarchs and how it's transmitted. I'll also just briefly touch on tropical milkweed, discuss then about fall migration and why it's important for monarchs' population health. And then finally, I'll tell you about a recent study I conducted with colleagues from UGA and Emory, looking at long-term patterns of infection in North America in monarchs. I'm leaving a lot of time for questions, so as they come up for you, please post them in the chat, and I will do my best to answer as many as I can at the end. Another thing I wanted to mention is that I will also be asking you some questions, so be ready to put some answers in the chat if you're comfortable and would like to participate. All right, so let's get started. So Ophryscystis electroscera, or OE for short, as I will refer to it, is a parasite of the monarch butterfly and also of the queens and some related butterflies. Parasites are organisms that generally weaken or harm the host when they grow on or inside the host. So the parasite is benefiting and the host is harmed in some way. So here I'm just showing you some pictures of the parasite if we were to zoom way in. And to me, these are the parasites that are really the most impactful for monarchs. Certainly there are other parasites and natural enemies of monarchs. To me, it also, these look like little deflated footballs or maybe boats. What's important to note is that these are actually spores of the parasite. And what's really neat about this particular protozoan is that it's easy to detect it. All we have to do is press a clear sticker onto the abdomen and look under a microscope. So no high tech is involved in determining whether or not a monarch is infected. OE is present in all monarch populations. So here's a map of the world, and in orange are areas where monarchs have been found. And in all of these populations, OE has been detected. Monarchs don't just occur in North America, where they're migratory. They're also found in Central and South America and the Caribbean. And at some point in 1800s, monarchs were introduced to Australia, New Zealand, Pacific Islands, and Bermuda. And again, in all of these places, OE is found. There's variation in the prevalence or how many of those monarchs are infected. But for today's talk, I'm really going to be focusing on North American monarchs, those that migrate. All right, so if we were to look at an infected individual, we were to zoom in into its abdomen, we would see these spores, these tiny little footballs, boats, whatever you like to think of them as. Again, important things, these are spores. They're on the outside of the body, and the monarch is covered with millions of dormant spores. Once infected, a monarch will never recover. It will always be debilitated in some way. Another important thing to say is that spores are not reproducing on the adult. They have to be consumed by a caterpillar to actually cause infection and damage. I like to think of OE spores as glitter, because if you've ever played with glitter for an art project, it gets everywhere. It sticks around. Months later, you're still finding glitter in some place. These spores are kind of like that. And this actually has a lot of implications for the parasite transmission in the wild, and also if you are rearing monarchs indoors. So something to keep in mind. A little bit more about the parasite. There are actually 3 ways that a caterpillar can become infected with the parasite. The best way that's been described is from an infected female to her egg. So the parasite is well linked to the life cycle of the monarch. And I think we're very familiar with the amazing transformation that goes from an egg to a caterpillar to pupa and so on. Well, when an infected female is laying her eggs, those spores are deposited from her abdomen onto the eggshell. The caterpillar then is emerging and eating that eggshell. And as soon as it is inside the gut of the caterpillar, that is when that spore will lyse and those parasites start replicating inside of the caterpillar. It's when it's replicating, that is when most of that damage is occurring. It continues from the caterpillar stage all the way through the pupal stage. Most of the damage is actually occurring in the pupal stage. And the adult that emerges is then covered with millions of dormant spores on the outside of the body. And as I mentioned that, those spores are kind of sticky like glitter. if you imagine an infected adult visiting a milkweed, some of those spores end up on the milkweed that it visits. This visit may be just resting, it may be nectaring, but nonetheless, some of those spores end up on the milkweed. And that means that if a caterpillar is from another individual on that milkweed, it will potentially ingest a spore and become infected. So milkweed can become contaminated with the spores. Another sticky situation is when adults are mating and there is transfer of those spores from an infected to a healthy adult. And again, those spores, just like glitter, will stick around. And so the healthy individual becomes a carrier. And let's say this is a healthy female that now has acquired some of those spores. She may be egg laying, and those spores end up on the milkweed and end up in her caterpillars. So we can have contaminated individuals as well contributing to infection. Lastly, I just wanted to mention that if you imagine a scenario where we have a bunch of infected individuals, the more of them we have visiting milkweeds, the higher the risk of infection for all monarchs that are utilizing the milkweed or a milkweed patch. All right, so what are the effects on the monarchs themselves? The answer is that it varies. So we can have a monarch emerging with all those dormant spores, but it appears normal. On the more extreme end, we could have a monarch that is very much damaged, it's wrinkled wings. And then on the most extreme end, we have a monarch that cannot close at all from its chrysalis. And most commonly, we really only see the normal looking infected monarchs. We don't often see wrinkled or unable to enclose monarchs because those die before most of us see them. The normal looking monarchs are nonetheless impacted. So there has been damage done to them during that pupal stage when those parasites were replicating. So they tend to have shorter lifespans, reduced mating success, and they don't fly as well. They're actually a lot slower flying, and they don't fly as long as healthy individuals. All right, so now I have a quick question for you. Have you seen wrinkled monarchs this year? And so that's the first one. And do you think OE is a problem where you live? And you don't have to disclose where you live, but do you think OE is of concern? So I will just give you 15 seconds here to answer the question, and then we'll move on. Next, I want to talk to you a little bit about tropical milkweed. So one of the questions I get a lot is, but isn't Asclepias curassavica tropical milkweed medicinal? And maybe if it is, we should plant more of it, because if OE is a problem, or we suspect that it's a problem, we can buy some at Lowe's or Home Depot, and it may solve some of the issue. So research does indicate that tropical milkweed, Asclepias curassavica, has a whole bunch of toxins in higher quantities than some of our native milkweeds that are native to North America. Not all, but most of them. And those toxins do play a role in that replication of the parasite inside the caterpillar and the pupa. So if we were to take an infected caterpillar and raise it on curassavica, when it emerges, the adult is still infected, but it actually has fewer spores. Maybe instead of 5 million, it has 3 million dormant spores on the outside, and it lives a little bit longer. Now, that may seem like, okay, that's actually good, but at the same time, that's not so good, because if an infected monarch is living longer, it means that it now has more time to spread the parasite spores onto milkweed or onto adults or so on. Another interesting thing that we have learned over time about tropical milkweed is that there's some preference for infected females to use Curasavica. So in an experiment where researchers put females, infected females, and gave them a choice to either lay their eggs on Curasavica or Incarnata, which is a native swamp milkweed with fewer of those toxins. And they compared that to uninfected females, so healthy females, and they had the same choice between those two plants. The infected females laid 70% of their eggs on this Curasavica plant, compared to uninfected females that kind of split their eggs 50-50, halfway. So it appears that there is a recognition by the infected female that there could be some benefit for her offspring to use this slightly more toxic milkweed so that their offspring do better, that the next generation isn't as impacted by the parasite. All right, so that is one of the really interesting things about tropical milkweed. Females that are infected tend to lay more eggs on tropical milkweed. But because of that, we also tend to see higher infection rates associated with this species, particularly in the southeastern US. And because this plant is non-native to North America, it's actually native to Central and South America, it does something a little different than our native milkweeds, in that it continues growing and flowering and looking really pretty until there's a freezing event. So it doesn't enter dormancy like most of our native milkweeds do, and that's actually quite important. And there are two reasons why that's important. So let me back up here first and explain why dormancy in milkweed is key. So most of our native milkweeds enter dormancy in the fall. So right around September, October in the northern areas of US, the native milkweeds will start to look a little yellow, and it's, not so great, and it really kind of dies back. And we don't really see a whole lot of caterpillars on them anymore. So as the days are getting shorter and the temperatures are cooler, monarchs are no longer utilizing the milkweed. They are also getting the message that, hey, winter's coming and we need to stop mating, stop reproducing, and it is time to head to an overwintering site. It's time to fly. Well, with a milkweed that is continuing to produce flowers and provide leaves for females to lay eggs on and for the caterpillars to munch on, they are not getting the message that it is time to fly. So some of my research during my graduate work indicated that monarchs which are raised on Curasavica plants compared to our native milkweed Incarnata are less likely to be ready to fly to an overwintering site. And for the southeastern US, it means that monarchs will actually continue using the milkweed and breeding into the winter months. And my former lab mate and friend, Dara Satterfield, did a lot of research looking at tropical milkweed. And in the southeast, in Georgia where we live, she found that tropical milkweed, Asclepias curasavica, was supporting caterpillars and supporting adults through the winter. She had also found that the infection rates at these sites were really high. So now I have a question for you. when was the last time you saw a monarch caterpillar on your milkweed? And when was the last time you saw an adult? So the adult could be just flying around. It doesn't have to be on milkweed, but I'm really curious, when are you seeing the last of the caterpillars, and are you seeing adults? So if you are comfortable, go ahead and include the state that you're in your answer, because again, I'm curious about the patterns that we're seeing this year in monarch reproduction. So I will give you about 10 seconds to go ahead and answer those two questions. All right, and I did forget to mention there is a little bit of a delay between when I am talking and when you're seeing this in the webinar. So I am not going to be able to see your answers right away, but I will be looking at them at the end. So I will definitely get to take a look at your answers at some point. Okay. So I wanted to mention that, so I am in Athens, Georgia, and I still have caterpillars on my milkweed, and I have some native milkweed. So they're definitely still trying to finish their life cycle. I'm not sure that they'll make it on time before there's a freeze, but they're around. And for this part of the country, we are now seeing waves of monarchs migrating through this area. And that is definitely my favorite time of year, when it's sunny and monarchs are flying around, and I'm wishing them best of luck to make it to their overwintering site. All right, so now I want to just touch upon the importance of migration for monarchs' population health. And I want to acknowledge a whole bunch of work that was done by my PhD advisor, Dr. Sonia Altizer. So for the past 20-something years, Sonia has been studying migration and the pathogen transmission in the Monarch OE system. And as you know, migration is a very long journey, and it's quite long for the eastern monarchs compared to the western monarchs. And I do want to acknowledge the fact that it is essentially the same population, but we do think of them as, ecologically they are a little bit different. And the journey for the eastern monarchs is quite long. To Mexico, it's a little bit shorter. To the coast of California, where they're overwintering. And Sonia's work has showed that migration greatly reduces parasite infection in monarch butterflies. So here's just a map of North America with some arrows indicating more or less the path that monarchs are migrating in the east and the west. And so there are two phenomena that are occurring. So first, migration allows monarchs to periodically leave behind that contaminated milkweed, where those infectious spores are hanging out, building up on milkweed plants. And so this is what we call migratory escape. So they're literally escaping infected environments. Second, if we were to think of ourselves as having to run a marathon with a flu, you can imagine that if you're infected with something, you may have to drop out of the race during, trying to make it really far, long distance. So it actually lowers infection rates through a process we call migratory culling. And there is actually really good evidence for this, not only in monarchs, but also in other migratory species. So this is a phenomena that is quite important across different systems. And some of Sonia's seminal work was back in the 2000s, where she had examined infection rates over the years for the eastern migratory population Western migratory population, and Florida resident population. And so in the southern tip of Florida, there are some monarchs that don't migrate. And those tend to have really high infection rates compared to the other migratory populations. And so it does appear that in east part of the country, the rates have been quite low for a while. And this would be consistent with a long journey, letting go of those infected individuals. And the longer the journey, the stronger the effect. So as I mentioned, in the West, it's not as long of a journey and strenuous as it is in the East. So there's been evidence for a while that this is occurring in monarchs. However, this paper was published in year 2000. So it's been a while since we have looked at the data and thought about it. A lot of things have changed since the year 2000. So the question that came up for me was, are we still seeing the same trends? Has anything changed? And so to answer this question, I decided it was time to do a new study, compile a whole bunch of data. And I worked with a dream team of monarch biologists and scientists. So Jaap de Rood from Emory University, who was at the time my postdoctoral advisor, Sonia Altizer from UGA, and Andy Davis. And we really asked, what are the long-term trends of OE infection since, how far can we go? I want to mention that this study has now been published in Journal of Animal Ecology. Here I just included the title in case you are interested. But I want to give you an overview of our findings and what we did and what we learned from all of that data. So first of all, we dug up a whole bunch of data that's been collected by researchers and that's been published previously. There were over 50,000 monarchs sampled that we included in our data set from Eastern and Western population. Here I'm just showing you a map of North America, the general region where Eastern monarchs were sampled with some gray dots indicated on the map and some on the Western. Just wanted to mention, it took me about a year to put all this data together, just because of the sheer number of different sources that I was looking through, and organizing all this data was quite a feat. A couple of important things I wanted to mention about this data. It only includes adult monarchs, so no larvae. It includes no citizen science, community science data. And we did not include any winter or resident breeding populations, right? So earlier I mentioned that, there is the Florida resident population. I also mentioned that there are in Georgia, some populations that continue breeding through the winter. None of that was included in this data set. So one of the things that we first asked was, are there differences in the Western versus Eastern population? And yes, we found there were differences. And this was not a surprise because there were trends that I just showed you before on the paper from the year 2000. So this was not a huge surprise. I do want to say that this includes data going back to the 1960s. We have a lot more data for Eastern North America than Western, but the pattern is consistent with what we have found before. Now, what about over time? When we plotted infection over time, we noticed that in Eastern Eastern North America, there's been a bump in infection since about the year 2000. And that's in the Eastern, but a very different pattern of infection in the Western. Again, the data is more limited for Western, but certainly interesting patterns that we're noticing. So here, the plot of infection rates over time. I want you to just notice that the Western data starts at about 1980, as opposed to the Eastern, which started about 1960. So slight differences there in how much data we have, which is important to consider. All right, so we noticed there were these differences. So we started asking, okay, what's going on here? Why are we seeing this? Is it because, we're lumping all this data into a whole year, but there are seasonal differences potentially? And I just told you that there was importance of migration in reducing infection rates. Are we missing out on some of that by just putting it all per year? And so first we asked whether or not we could detect this migratory culling, or the loss of the infected individuals. So when we looked at the data by the breeding season, migratory season, and overwintering, we saw that the proportion of infected adults in the Eastern US did differ between those. So during the breeding season, infection rates were about 5%, let's say, and then it actually increased during migration, which is something we did not expect, but then decreased for the overwintering monarchs that were sampled. So this would indicate to us that there may be some migratory culling, but it's not as strong as we expected it to be. But again, we have all these years lumped together. For the Western monarchs, we saw a much stronger trend for the decrease from breeding to migrating and overwintering population samples. So this indicated to us that there may be some culling of the infected individuals, but maybe we weren't seeing the whole picture because, again, we were putting all the data together, all the different years, and each year is a little different. So we thought it would be better for us to examine some of this information per year basis. So another way to think of whether or not we can detect migratory culling was to ask whether there would be whether or not there is a relationship between the proportion of infected monarchs and abundance estimates, or like how many monarchs were observed, right? Because if there is culling and monarchs are lost, you would expect that there would be some decrease. So in terms of estimates of monarch population, none of these are perfect, but there are two really main ones that are available, and that is overwintering estimates and summer abundance estimates. And so we decided that we would take a ratio of these two to ask, well, for a given year, are we seeing changes and losses of monarchs between those two estimates? Because a high ratio would suggest that summer monarchs are being lost. So that to us was an indicator potentially of culling. So then when we plotted proportion of infected adults against this ratio that I just mentioned, we noticed that there was, in fact, a negative trend. So proportion of infected monarchs was increasing when overwintering populations were lower in relation to summer populations. And the same trend was noticed for the Western monarchs. So that indicated to us that in the years that migratory culling was occurring with higher infection prevalence, we saw these patterns. All right, so we wanted to dig further, and this is the last result I'm gonna tell you about, but basically we wanted to know if there were any other factors that could be impacting the trends that we saw. So we asked whether or not climate was impacting infection trends, whether summer abundance, as NABA estimates could tell us, are impacting infection trends. Also, we were curious about greenness of the landscape, which would indicate resources, and lastly, breeding season length. And so here I am showing you it's the same data I showed you earlier, but now I have changed my y-axis. So over time, since the 1960s, this is the Eastern population infection rates. And as you can see, over time, it has increased from nearing 0 to 15% over the last 10, 15 years. And when we did some fancy statistics and we included all of these factors that I just mentioned into our analysis, the only thing that came up as having an impact on infection trends was summer abundance estimates. And I wanted to mention that these were for a particular region. So in a given region, the number of monarchs was positively related to infection trends, which makes sense given the dynamics of the parasite and how it's transmitted and how it's spread. For the Western monarchs, we again saw this decrease in infection rates over time. And again, we saw an impact of summer abundance. And surprisingly, we saw that breeding season length was negatively related to infection trends, which was puzzling to us. I think that one of the possibilities there is that the shorter the season is potentially clustering more monarchs and causing more spread of the parasite. Lastly, I just wanted to mention that I told you that there are contaminated adults in the population, and we saw trends that were similar to the truly infected, highly infected adults. Contaminated adults are out there in the population, and they are in the same proportion, Eastern versus Western relatively to each other. So it's much higher in Western compared to Eastern. Reasons why monarchs can become contaminated are various, but I just thought it was an interesting result because in the past we've really overlooked infected monarchs overall. Sorry, contaminated monarchs compared to infected monarchs. All right, so in summary, OE is this very specialized parasite that varies in severity. Infection rates have been changing over time, seasonally and also over the years. In general, in the Eastern US, we have seen an increase in infection rates since the 1960s, nearly tripling in some years, which is causing some concern. And migration is really key to reducing infection rates. Also, it means that if we have a lot of infected monarchs, a lot of those infected monarchs are being lost during migration season. And it's a possible explanation why we're seeing fewer monarchs in overwintering sites if they are lost during the long journey. Lastly, I want to say OE is here to stay, and we need to continue monitoring it through one of the main ways that I would encourage you to contribute to monitoring is through Monarch Health, which is a community science project that Dr. Sonia Altizer has been leading for the last 15 years. And that is one great way to contribute to continuing learning about the parasite. All right, so with that, I want to show you this beautiful picture of monarchs overwintering in Mexico. this is the time of year when I think of this the most. They are making their way down, but if they are infected, they're not going to make it. So we need to continue to keep an eye out on this. And with that, I want to thank you so much for joining me today. Please email me with questions that I don't get to answer today. My email is just my last name at uga.edu. I want to thank Monarch Joint Venture and for the invitation to do this webinar today. And also I need to acknowledge my funding sources, which included Emory University and NSF. All right, so with that, I am going to take some questions.
Awesome. Thank you so much, Ania, for sharing your knowledge with us today. We are going to take the next few minutes for Q&A. So if you still have questions, go ahead and put those in the chat. If we don't have time to answer every question, maybe we'll see if Ania would be willing to answer them, and we can stick them in the follow-up email that we'll send out later this week. Okay, first question, Ania. This one isn't necessarily related to OE, but you did mention monarch populations on other continents, and somebody was wondering if those populations migrate in the same manner that the North American monarch populations do.
Great question. There is some indication that in Australia they're doing a very short migration, but nowhere near what we're seeing in North America. So for the most part, the answer is no, but there's definitely, it seems like there is something left in the genetic makeup of the monarchs that wants to continue migration. But the short answer is generally no.
Right. Okay. A couple of people actually were wondering if OE spores can be rinsed off the milkweed with a garden hose.
Ooh. Well, so in the lab, whenever we are bringing milkweed from the greenhouse or from outside, we always rinse the milkweed, but not just with water. We've also rinsed them with just a little bit of bleach just to get those spores dislodged, just as the same way as glitters just really sticks. There's, I think, a little bit of like static electricity that helps the spore to hold on to the surface. So I think that it's not been tested officially, but I do think that you could probably get some spores off, but to really truly get them all off, you'd probably have to power wash or do a bleach wash of the milkweed, which will have some impact, on the plant. So not what I do.
And I would imagine a power wash would kind of pulverize the leaf. Okay. Could you talk a little bit about what the origin of OE is? Like how did it start infecting monarchs? Do we know anything about the evolutionary history of OE and monarchs together?
Well, that's a great question. So I don't know that anybody has explored or dug into, trying to figure out when was the first infection. I think that would be really hard to answer. As far as we know, it has, co-evolved for quite a long time with the monarch. That's why it's so specialized. And so I am just going to take a wild guess here about the fact that how it evolved. It could have been a protozoan that, maybe was of another species or some other insect that a caterpillar had ingested. And by chance, at some point, those spores were successful at infecting the gut of the caterpillar. And that's how it probably initially started. But this is just a speculation. I don't think there is actually any work. As far as we know, it's been around for millennia with the monarch, and yeah, that's it.
Co-evolved. Yeah.
Evolved, yes.
Yeah. Okay. I'm going to jump around some of the questions here because I know that we've got lots of time, but I want to make sure that we're getting to the ones that I think will be most beneficial for everybody who's asking them. Could you talk a little bit more how those of us who are participating in community science can check for OE, or where we can go to read further about it. And we'll share these resources in the follow-up email just so everybody knows.
Sure. I think that if anybody just Googles Monarch OE parasites, the first page that's going to come up is Project Monarch Health. That's at the University of Georgia with Sonia Altizer. And it will bring you up to more information on how you can request a kit to sample adult monarchs for infection. And so it is one way for you to do it is to request to participate in the project. You can also purchase little mailing clear stickers and note cards, because that's all it is a note card and some clear stickers to sample monarchs. Definitely want to be wearing gloves whenever you're sampling adults. And you just want to be cognizant of that glitter effect, that if you are sampling monarchs, that it will get everywhere. So if you are, for example, running around outside with a net catching them, your net will also at some point be contaminated with the spores if you are catching infected monarchs. So you want to make sure you are doing your due diligence and washing your equipment afterwards, wearing gloves, and trying to keep your work environment as clean as possible when you are sampling monarchs. So changing gloves, and you won't become infected with the protozoan, but washing hands is always just a really good practice. I think that there is also a possibility of, if you, let's say, have a 5th instar in your garden, you can collect it to sample, but then they have to go through the pupa stage, and then you sample the adult once it emerges.
And like I said, we'll share some resources with that. All of the Monarch community science programs talk to each other. And so Project Monarch Health and the Monarch Lever Monitoring Project both have not necessarily shared protocols, but shared resources for how to participate in one or the other. So we'll they go well together. So we'll share that in the follow-up email also. Ania, there is a question about why there are fewer studies being done on Western monarchs and OE, since it seems like that population has a more severe decrease. Could you talk about that for just a second?
Yeah. Well, I think one of the challenges with studying Western monarchs is that there are just fewer of them. So that's for the start. When it comes to bigger sample sizes, Eastern population is a lot easier to work with. I think that perhaps because the long distance spectacular migration is really part of the Eastern population, maybe there hasn't been just as much interest in the Western monarchs. There's definitely a lot of gaps in our understanding about Western monarchs, and I agree that there needs to be more. But I want to encourage people to study Western monarchs, and I do think that there are some specific community science programs that are just for the Western population. I just don't think that they are as well known. So they have to do something.
And we can share those resources as well. I'll add that to our document. There's the Western Monarch Milkweed Mapper, and then the Western Monarch Thanksgiving Count, which looks at overwintering monarchs. But I'll add to that too, Ania, if you don't mind. They're currently in California, you need a permit to handle or collect monarch butterflies, which is also another challenge. And they're not issuing very many of them at all right now, just because of the concerns with the population. So just be aware of that. Currently, unless you have a permit, you cannot test for OE on monarchs in California. So Ania, do you want to check the chat real quick? I just dropped a couple of things in there. And then I'm going to scroll through some questions here. Somebody was wondering, kind of related, are you seeing OE in the population of monarchs in Mexico? I don't know if they mean specifically overwintering monarchs or other non-migratory monarchs. And then somebody was also wondering about Hawaii and if how those results in Hawaii are accounted for.
Yeah. So yes, so there are monarchs that are infected that do make it to Mexico. They do make it all the way to the overwintering sites. And actually, that is, if you think about from the parasite perspective, it's actually really important that there are some individuals that make it, right? The parasite doesn't want to entirely kill off its population because then also it will go extinct and die. So that's not ideal for the parasite. So some infected monarchs make it because those monarchs are like tightly clustered on those trees. There is some of that transfer between adults, and later in the spring when those monarchs are mating, also there could be transfer between infected and uninfected adults. As many as 1,000 spores can be transferred between infected and uninfected monarch. So there are definitely there. It again tends to be a little bit lower just because so many of the infected ones were lost during migration. When it comes to resident monarch populations such as Hawaii and Bermuda, any of the islands, the infection rates tend to be a lot higher. And, we're talking anywhere between 30% to 100% of monarchs at a given site. So it definitely varies. Some of the work that I had done during my PhD indicated that even where the milkweed is located, whether it's a more urban or rural area, can dictate the infection rates. And I think that really has to do with availability of the milkweeds and how many monarchs will visit a single milkweed plant. If you imagine that, there's only, let's say, 1,000 plants available, and that's all that is, all the monarchs will go to those 1,000 plants, and infection can spread really quickly between sites, especially if they're mating and transferring those spores to each other and then moving on to another patch. So those tend to be high, perhaps because the monarchs really don't have anywhere else to go.
Great, thank you. There is another question that I can address real quick here while you read the chat, Ania. Somebody was wondering if cutting back tropical milkweed in regions that don't freeze, if that's still recommended. And as far as my knowledge is, it is still recommended to cut back tropical milkweed in regions where it does grow year round. Typically that's, you want to start cutting it back right around now in October and continue cutting it back through February or early March, just to kind of mimic what the native milkweeds are doing and going dormant. And that will help cut back on some of those infection rates and discourage winter breeding in the monarchs that are passing through there. Mm-hmm. And then another question, Ania, you could address this one too, is OE only, does OE only infect monarchs, or can it infect other species of butterflies as well?
Yeah, so it can also infect queens and soldiers, and it's been detected in related Danaid butterflies. So there are also OE-like parasites in some of the related butterflies in Africa. So the parasite is definitely specialized in the genus, but for example, monarch cannot infect a black swallowtail. because partly, well, it just doesn't, I'm going to guess the parasite does not lyse correctly and replicate inside the caterpillar. Diet is different environment for the parasite, and so it doesn't there's no cross species infections. I also wanted to address the question that I saw in the chat regarding infection rates at overwintering sites in California. So the question was, why is the OE infection rate in overwintering site in California nowadays around 5% versus 30 to 60 in the 80s and 90s? So this really has to do with abundance of the monarchs. So we call the OE parasite density dependent, meaning that you need essentially the higher the abundance and density of monarchs, the higher the risk of infection. So 40 years ago, when we had a lot of monarchs in California and overwintering sites, then the overall population was larger, and the infection rates were therefore higher compared to nowadays, where we have such few monarchs in the California area, where the transmission is really not occurring as commonly, because there are just fewer monarchs around to infect each other.
Wonderful. Thank you.
Yep.
All right. So we've got a couple more minutes. This is an interesting one. I don't know if I've ever had anybody ask this one before. Are there any natural predators for the spores or for the OE parasites?
That's a really good question. No, as far as I know, no. As far as I know, it's but I'll tell you something. I have some data that I haven't published yet from my PhD work, where I noticed a pattern between the number of aphids on the milkweed plant and the probability of infection for a caterpillar. So the more aphids there were, the less likely any caterpillar would get infected. And I think it's because the aphids are essentially picking up some of those spores when they're walking around the milkweed, and they're essentially pulling them away. That doesn't mean that they are completely, taking away all the spores. Mm-hmm. Lots of monarchs are still becoming infected, but the probability is just a little bit lower, which I thought was really interesting. Aphids will impact the milkweed quality. So there is also a flip side of that the quality of the food for the monarch is not as good. So there could be some effects on size and so on. But there is, as far as I know, besides aphids potentially moving around the spores, and a freeze just, killing the milkweed and taking it essentially away, there really aren't any natural enemies. I think time is an enemy that kills us all, but
time gets us all.
Yeah. Gets us all. But that's the main one. Yep. Cold and time.
Cold and time and bleach.
Bleach. That's true. Yeah. So it's not.
All right. Well, we are just about to the hour, so I think we will wrap it up there. There were a few more questions that we didn't get to today, so Ania, I'll send you a follow-up email if you have time to answer them before we send out our follow-up email on Friday. But for those of you who have registered and we have your email addresses, then you'll receive that follow-up email by the end of the week with the resources and any unanswered questions that we can get to before then. But I do want to say thank you again to Ania for presenting today, and thank you to NCTC for hosting again, and to all of you for participating. We're so happy to have you with us. We hope you'll join us for the next webinar, which will be one will be joined by Dr. Cuauhtemoc Saenz Romero and Dr. Jose Arnulfo Blanco Garcia on November 14th to present about the climatic change impacts on the monarch butterfly biosphere reserve, Aves religiosa, the sacred fir forest. please note that the next webinar will be on the second Tuesday of November, not the usual fourth Tuesday. Like most months, there is a major holiday in the United States that we have to work around. So the date of the next webinar will be November 14th. We'll be at the same time, same place. So watch your emails for the reminder, and we'll see you all next time. Thanks, everybody.
Thank you.